Welcome to the course on biostatistics and design of experiments. We are talking about ANOVA, two-way ANOVA, and the most important point which I introduced in the previous situation is interaction between two variables or two main effects or two independent variables. So, if it is not an additive, suppose when I am changing temperature and when I am changing pH, at one particular value of pH it appears to go like this, in another value of pH instead of going almost parallel it seems to be sort of converging. So, obviously there is an interaction between pH and temperature, okay. Or you may have a situation like this, at one pH the yield goes up as I change temperature, but at another pH yield is dramatically rising. Obviously there is an interaction between pH and temperature, okay. So, interactions are very important, I said, and we need to study interactions, and only when you do replications you will be able to study interactions. Without replication of data you will not be able to study interactions. Okay, let us look at one more problem, okay, which again looks at a two-way ANOVA, okay. It is called a two-way ANOVA. Okay. So, here in this particular problem we are looking at the production of a quorum sensing molecule. Quorum sensing is a very important concept in bacterial biofilm formation, okay. So, the bacteria goes and attaches to surfaces, and it could be any surface, it could be a inert material, synthetic material, biological material. So, when the bacteria settles and it produces EPS and proteins, carbohydrates, they form a layer which is called biofilm, and biofilm is very important for the growth of microorganisms and so on actually. And a molecule AHL, acyl homoserine lactone is involved in identifying the quorum, okay. So, bacteria produces this AHL, and when there is sufficient amount of AHL that is observed by the bacteria, they know that the amount of bacterial is high. So, they go into a different phenotype and they start forming biofilm. So, that way it is called a signaling molecule AHL, okay. And AHL has many implications. So, if I can prevent AHL, I may be able to prevent the biofilm formation. The AHL of one bacteria could be toxic to another bacteria. So, that is more like a defense mechanism and so on actually. So, there is some interest in looking at bacterial AHL production and use this as an antibacterial compound and so on actually, okay. So, production of AHL, this quorum sensing molecule, is being optimized using 4 different media A, B, C, D, and 5 different organisms. Many organisms produce, as I said, lactones are formed as AHL signaling molecule. In some cases there are peptides that are formed, okay. So, and then even in the lactones there are different types of lactones that are produced by these organisms. So, imagine you are testing 5 organisms and 4 different media for AHL production, and you get you are doing a replicate. So, obviously you have 2 sets of results, the amount of AHL produced in milligrams per liter, okay. So, now you have been asked to perform a two-way ANOVA. So, basically you have 40 data points. So, totally 39 degrees of freedom, okay. Now, let us put it down in the usual form. So, we have the replicate data also here, replicate data for each one of the organism also here, okay. So, we have totally 40 data points. Now, we can get a media average, just take the average of all these columns, that is called the media average. So, for media A the average comes out to be 20.1, media B 19.4, media C 22.1, media D 18.1. Similarly, for organism also we can get an average if you go this way. Again 10 data points for organism 1, 10 data points for organism 2. So, we get the average for organism. So, we can get the media average, we can get the average for organism, we can get the global average, that means average of all averages. So, we get the global average is 19.925, Okay. Now I need to find out media sum of squares. You all know how to do it, right? You have done it now many times. So what do I do? So I have average for each one of the media. So what do? I subtract from this global, square it up, okay, and multiply by 10. Why? Because there are 10 data points here. 10 into 20.1 minus 19.925 square, okay, 10 into 19.4 minus 19.92 square, and so on. So for the media you get 4, you add up all of them, that gives you media sum of squares, understand. So that is called the media sum of squares. Now let us go for organism sum of squares. So you have organism average here, okay, 5 different averages, okay. So what do? You take 19.75 minus global, square it up and multiply by 10 because you have 10 data points, simple, right? That is what I am doing here, 10 into 19.75 minus 19.925 and so on. So here 10 into 18.5 minus 19.25 square, okay, and so on. 10 into 19.375 minus 19.25 square, 10 into like that, 22.62 minus 19.9 square, 10 into 19.37 minus 19.9 square. So for organism you will get 5, you add up all of that, that will give you a organism sum of squares, okay. So we get media sum of squares, we get organism sum of squares. Now total sum of squares, you have to take the sum of squares with respect to the x double bar. The global average is called the x double bar, right. So what do? 20 minus 19.925 square, like that, 20 minus 19.925 square, 18 minus 19.925 square, 20 minus 19, like that, you get this entire exactly here, and then you add up all of them, that will give you the total sum of square 196.775. Okay, now how do I get the error sum of squares? Now you have every time you have replicated, so obviously this is an indication of the error, right. So if you take an average and then the change difference from the average, square it up, that is an indication of the error. So then you can add up all of them, okay. So again I am showing the same data here. If I take the average of 20 is 20, 18, 20 is 19, okay, and then 19, 19 is 19, 22, 24 is 23, 19, 20 is 19.5. Similarly here 19, 20 is 19.5, okay. So you will get 5 data set, okay. These are average of each one of these groups. So how do I take the sum of squares? So 20 minus 20 square plus 20 minus 20 square, 18 minus 19 square, 20 minus 19 square, 19 minus 19 square, 19 minus 19 square, 22 minus 23 square, 24 minus 23 square, like that, because these are indications of the error, because these are deviations from the mean. So you have this box where you have repeated, and the deviation from the mean of this each group is the repeat sum of squares or error sum of squares, and if you add up all these you will get the error sum of squares here, you understand. So we get the total sum of squares, you know how to do the total sum of squares, you are subtracting from the global average, you know how to do the media sum of squares, because we have 4 media, each media will have its own mean, you subtract from the global, square it up, multiply by 10 because 10 data point. Now media sum of squares, organism sum of squares, for each of the organism you get an average. In this case there are 5 organism, so each of these you subtract from the global, square it up, multiply by 10. Why 10? Again there are 10 data points in this particular situation, so you add up all of them, that will give you the organism sum of squares. Total sum of squares, you subtract each one of the term with the global average, square it up, then add whole lot. Okay, now how do you get the repeat? You have done it twice, each experiment is done twice. Obviously there is an average, and the difference between the average and individual value square is a measure of variance of that particular box, understand. I will call this a box, each one is a box, okay, each one is a box here. Now you add up all of them, that will give you the error sum of squares. So we have the total sum of squares, we have the media sum of squares, we have the organism sum of squares, we have the error or repeat sum of squares. So if you add all these 4 and subtract from total sum of squares, that should give us interaction sum of squares, okay. So we have 4 media, so we will get 3 degrees of freedom, okay, and media sum of squares, as you see, 83.675, so we put that as 83.675. There are 5 organisms, we have 4 degrees of freedom, organism sum of squares is 79.65, okay, 79.65, total is 196.775, 196.775, degrees of freedom is 39 because we have 5 organism and 4 media. So 5 into 4 is 20, repeated each one twice, so 20 into 2 is 40, 40 minus 1 is 39. So organism into media, you get it as 3 into 4, 12. Now error sum of squares, we got it as 21.5, 21.5. We know total 39, media we know 3, organism is 4, organism into media is 3 into 4, 12. So 12 plus 4 is 16, 16 plus 3 is 19, 13 minus 19 is 20, okay. This is how we calculate the degrees of freedom. Now each of the sum of squares we divide by the degrees of freedom to get mean sum of squares, 83.6 by 3, 79.6 by 4, 11.9 by 12, 21.5 by 20. You understand? Now how do I calculate f value? Here I am introducing something different. I am talking about fixed effect model and random or mixed effect model. So we have been all the time dividing by error, right, that is called a fixed effect model. So 27.89 by 1.1075, 19.9 divided by 1.075, 0.995 divided by 1.075, okay. So these are the f ratios. Now in the random or mixed effect model, what do you do is, okay, the interaction is divided by error, but main effects are divided by interaction. So in the random or mixed effect, 27.89 divided by 0.99, 19.91 divided by 0.99 and 0.99 divided by 1.075. So there is a difference between fixed effect model and the random effect model. In the fixed effect model, you divide every MSS with the error, whereas in the random effect model only the organism media, that is the interaction effect, you divide by error, whereas the main effects are divided by the interaction effect. Okay, now we go to the F table, we have 3 and 20 degrees of freedom, or 4 and 20 degrees of freedom for 0.095, 3 and 20 degrees of freedom 3.1, okay. Okay, 4 and 20 degrees of freedom, 4 and 20 degrees of freedom is 2.87, okay, 2.87, or even 12 into 20 degrees of freedom. Next page, 12 into 20 degrees of freedom is 2.828. These are actually valid for fixed effect model. For random effect model, I will be it is not 3 into 20 degrees of freedom, it will be 3 into 12 degrees of freedom, because in random effect model you are dividing by the interaction. So it will be 3 into 12 degrees of freedom. So in 3 into 12 degrees of freedom, if you are looking at, you will use 3.49, okay. So this F table for random effect model will be 3.49, and 4 into 12 will be 3.26, okay, 3.49 and 3.26, okay. Do you understand? Because your degrees of freedom has gone down, so 3.26 and 4.2, okay. So if you look at this say fixed effect model, you see that the F value which you calculate from ANOVA as against the table value. So media has a very strong effect, Okay, so obviously there we take the alternate hypothesis media has a strong effect, and then organism also has a strong effect here, okay, whereas interaction does not have any effect, whether it is a fixed effect or the random effect model. Okay. So out of these so if you look at all these data, so we have these 4, okay. We have these 4 different media, we have these 5 different organism. So this particular media C seems to be producing largest amount of this particular molecule, and similarly organism 4 seems to be producing largest amount of this molecule. So as you can see, it is very difficult to use 2 sample t test. So we have to solve it by ANOVA, and ANOVA also gives this 2-way ANOVA. We are looking at the main principal effects are the organism and media, and the interaction which we saw, that is the interaction between the organism and media. In this particular case we do not see any interaction because this calculated value is much smaller than the table value, understand. So if you plot all these data, you see that different media, different organism 1, 2, 3, 4, 5, so they are almost like parallel, you know. So they are not crisscrossing or going up and down. So all the organisms approximately perform in the additive way towards these media. Okay. If you compare this particular graph as against your previous problem, if you remember, okay, they were going almost like this, you know, 2 drugs and male and female. THZ is performing like this, whereas your metformin was performing like this. So metformin was performing much higher effect on female when compared to on the male. So both the drugs are almost similar, little small differences on male, whereas metformin was performing very differently when compared to THZ on female. So there was an interaction, the lines were not parallel, whereas in this particular case you see that the lines are parallel, almost parallel. This is the organism 1, 2, 3, 4, 5, and these are the different media, as you can see here, different media. So the effects are additive between the media and the organism, and it is not a nonlinear type of dependence or an interaction type of dependence. Okay. Now the important point here is we had enough data sets, so we are able to understand interaction also. Suppose I did not do a replication, okay, I did only one experiment in each case. Okay, what happens? So I have done only one experiment in each case, I did not do a replication. Okay, so total number of data points are 20, so my degrees of freedom for total is 19. Media, there are 4 media, no change, so degrees of freedom is 3. Organism 5, organism degrees of freedom is 4. So if I want to study organism into media, 4 into 3, 12. So 12 plus 4 is 16, 3, error becomes degrees of freedom of 0. That means I cannot understand error. Okay, so the interaction should be called as error, then it becomes easy for me to calculate f value. So do you understand? So interaction gets mixed up with error, or interaction gets confounded with error. I am introducing a new term called confounding. So interaction becomes confounded with error, so we cannot separate out error and interaction, that is media and organism, because we do not have, if I take the interaction as 12 degrees of freedom, that is 3 into 4, error becomes 0 because you do not have enough degrees of freedom, or you call this error, then you cannot study the interaction. Okay, say either you call this error, then you cannot study interaction because you have only 19 degrees of freedom. Okay. So if you repeat the whole lot, then like in the previous case I showed you, okay, if you repeat the whole, what we have? We have 40 data points, so obviously your total is 39, media remains same 3, organism remains same 4, organism into media interaction becomes 12, so obviously error 12 plus 4 is 16 plus 3 19, 39 minus 19 is 20, you understand. So this problem also shows you the importance of replicating the experiment. If you do not replicate, it is not possible for you to look at interaction effects. Okay. So, you need to always replicate your data so that you can study the interaction effect also, otherwise interaction effect gets confounded into error. You will not have a sufficient degrees of freedom to perform these type of calculations, and that becomes a big problem actually. So, we cannot talk about interactions at all. Okay. So, we looked at different types of ANOVA so far. We looked at one way ANOVA, then we looked at two way ANOVA, two-way ANOVA with replication, without replication, then we looked at two-way ANOVA with interaction, that means the principal effects are getting non-linearly associated with each other. It is not a simple additive type of relationship, but it is a non-linear relationship. That is called an interaction effect. In some cases the interaction could be positive, that means x1 x2 could be positive, in some cases x1 x2 could be negative also, both the possibilities are there, and that is where interaction plays a very important role. If you want to really find out effects for interaction, generally what do we do? If we have the effect principal effect 1 certain degrees of freedom df1, principal effect 2 certain degrees of freedom df2, the interaction effect will be df1 into df2. Okay. So, when I am calculating the error degrees of freedom by subtracting all these degrees of freedom from the total, I should have at least 1 or 2 degrees of 1 degree of freedom for error, then only I can do a F calculation, because the sum of squares of error, mean sum of squares of error comes in the denominator. So, I should have some number, and if I want to calculate mean sum of squares for error, obviously I should have some degrees of freedom, at least 1 degree of freedom, otherwise it is not possible to calculate mean sum of squares for error. So, replication is a must. I should have enough data points so that I get degrees of freedom for each one of these terms. Okay. And that is why I showed, if I am doing it twice, each of the experiment, I get sufficient degrees of freedom for error, whereas if I am doing only once, then obviously I am in big trouble. I am not able to get any degrees of freedom for error, as I am showing here actually. Okay, in this, as show it in this particular example. So, obviously replication plays a very important role, and another thing is the higher the number of degrees of freedom for error, because when you calculate the mean sum of squares of error, you are dividing by the degrees of freedom. So, the mean sum of squares of error becomes very small. Okay. So, when that number becomes very small, that goes into the denominator when you are calculating F ratio. So, obviously the F values become very large. If the F values become very large with respect to the table value, then we can say that there is a statistically significant difference. That means we will be able to reject null hypothesis. If your degrees of freedom for error is very small, because it comes in the denominator for mean sum of squares for error, that term becomes large, and again that comes in the denominator when you calculate F. So, your F values become small. So, obviously those F values will be smaller than the table F value. That means you will not have any reason for rejecting the null hypothesis. So, you will not be able to see effects of various factors, whether it is drugs or whether it is fermentation pH or fermentation temperature or carbon amount and so on, if your degrees of freedom for error is not sufficiently large, or if you have too much of variations when you do a replicate experiment. So, in these two situations you will not be able to see effect of various factors or effect of various independent variables. Okay. So, you need to keep these points in mind. So, when I am doing a replication, I should be very sure that the variances or errors involved are small, and when as many replications are always good, because my degrees of freedom increases, that means my degrees of freedom for error increases, that means mean sum of squares for error decreases, that means my F values will increase, and definitely it will be much higher than my table value. Okay, thank you very much. So, we will continue in the next class further concepts about the biostatistics. Thank you for your time. Thank you.